Dear viewers, this video is part 4 of the 4-part series on the genetic control of cell function in the Medicine for Common Man series. Figure 17 shows the stages of cell reproduction. The 4 stages of mitosis are prophase, shown in figure 1.7 A, B and C, prometaphase in figure 1.7D, metaphase in figure 1.7e, anaphase in 1.7f, and telophase in 1.7g and h. Cell mitosis. The actual process whereby the cell splits into two new cells is called mitosis. Once each chromosome has been replicated to form the two chromatids, mitosis follows automatically within 1 or 2 hours in many cells. Mitotic apparatus. Function of the centrioles. See figure 1.7 shown earlier. One of the first events of mitosis takes place in the cytoplasm in or around the small structures called centrioles during the later part of interphase. As shown in figure 1.7, two pairs of centrioles lie close to each other near one pole of the nucleus. These centrioles, like the DNA and chromosomes, are also replicated during interphase, usually shortly before replication of the DNA. Each centriole is a small cylindrical body about 0.4 micrometer long and about 0.15 micrometer in diameter, consisting mainly of 9 parallel tubular structures arranged in the form of a cylinder. Two centrioles of each pair lie at right angles to each other. Each pair of centrioles, along with attached pericentriolar material, is called a centrosome. Shortly before mitosis takes place, the two pairs of centrioles begin to move apart from each other. This movement is caused by polymerization of protein microtubules growing between the respective centriole pairs and actually pushing them apart. At the same time, other microtubules grow radially away from each of the centriole pairs, forming a spiny star called the aster in each end of the cell. Some of the spines of the aster penetrate the nuclear membrane and help separate the two sets of chromatids during mitosis. The complex of microtubules extending between the two new centriole pairs is called the spindle, and the entire set of microtubules plus the two pairs of centrioles is called the mitotic apparatus. So, dear viewers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Prophase. The first stage of mitosis, called prophase, is shown in figure 1.7 A, B and C. While the spindle is forming, the chromosomes of the nucleus, which in interphase consist of loosely coiled strands, become condensed into well-defined chromosomes. Prometaphase. During the prometaphase stage, see figure 1.7D, the growing microtubular spines of the aster fragment the nuclear envelope. At the same time, multiple microtubules from the aster attach to the chromatids at the centromeres, where the paired chromatids are still bound to each other. The tubules then pull one chromatid of each pair toward one cellular pole and its partner toward the other pole. Metaphase. During the metaphase stage, figure 1.7e, the two asters of the mitotic apparatus are pushed farther apart. This pushing is believed to occur because the microtubular spines from the two asters, where they interdigitate with each other to form the mitotic spindle, push each other away. Minute contractile protein molecules called molecular motors, which may be composed of the muscle protein actin, extend between the respective spines and using a stepping action as in muscle, actively slide the spines in a reverse direction along each other. Simultaneously, the chromatids are pulled tightly by their attached microtubules to the very center of the cell, lining up to form the equatorial plate of the mitotic spindle. Anaphase. During the anaphase stage, see figure 1.7f, the two chromatids of each chromosome are pulled apart at the centromere. All 46 pairs of chromatids are separated, forming two separate sets of 46 daughter chromosomes. One of these sets is pulled toward one mitotic aster, and the other is pulled towards the other aster, as the two respective poles of the dividing cell are pushed still farther apart. Telophase. In the telophase stage, see figure 1.7 G and H. The two sets of daughter chromosomes are pushed completely apart. Then the mitotic apparatus dissipates and a new nuclear membrane develops around each set of chromosomes. This membrane is formed from portions of the endoplasmic reticulum that are already present in the cytoplasm. Shortly thereafter, the cell pinches in two, midway between the two nuclei. This pinching is caused by the formation of a contractile ring of microfilaments composed of actin and probably myosin, the two contractile proteins of muscle, at the juncture of the newly developing cells that pinches them off from each other. Control of cell growth and cell reproduction. Some cells grow and reproduce all the time, such as the blood forming cells of the bone marrow, the germinal layers of the skin, and the epithelium of the gut. Many other cells, however, such as smooth muscle cells, may not reproduce for many years. A few cells, such as the neurons and most striated muscle cells, do not reproduce during the entire life of a person except during the original period of fetal life. In certain tissues, an insufficiency of some types of cells causes them to grow and reproduce rapidly until appropriate numbers of these cells are again available. For example, in some young animals, 7/8 of the liver can be removed surgically, and the cells of the remaining 1/8 will grow and divide until the liver mass returns to almost normal. The same phenomenon occurs for many glandular cells and most cells of the bone marrow, subcutaneous tissue, intestinal epithelium, and almost any other tissue except highly differentiated cells such as nerve and muscle cells. The mechanisms that maintain proper numbers of the different types of cells in the body are still poorly understood. However, experiments have shown at least three ways in which growth can be controlled. First, growth often is controlled by growth factors that come from other parts of the body. Some of these growth factors circulate in the blood, but others originate in adjacent tissues. For example, the epithelial cells of some glands, such as the pancreas, fail to grow without a growth factor from the underlying connective tissue of the gland. Second, most normal cells stop growing when they have run out of space for growth. This phenomenon occurs when cells are grown in tissue culture. The cells grow until they contact a solid object and then growth stops. Third, cells grown in tissue culture often stop growing when minute amounts of their own secretions are allowed to collect in the culture medium. This mechanism too could provide a means for negative feedback control for growth. Figure 1.8 shows control of cell replication by telomeres and telomerase. The cell's chromosomes are capped by telomeres, which, in the absence of telomerase activity, shorten with each cell division until the cells stop replicating. Therefore, most cells of the body cannot replicate indefinitely. In cancer cells, telomerase is activated and telomere length is maintained so that the cells continue to replicate themselves uncontrollably. Telomeres prevent the degradation of chromosomes. See figure 1.8 shown earlier. A telomere is a region of repetitive nucleotide sequences located at each end of a chromatid. Telomeres serve as protective caps that prevent the chromosomes from deterioration during cell division. During cell division, a short piece of primer RNA attaches to the DNA strand to start the replication. However, because the primer does not attach at the very end of the DNA strand, the copy is missing a small section of the DNA. With each cell division, the copied DNA loses additional nucleotides from the telomere region. The nucleotide sequences provided by the telomeres, therefore, prevent the degradation of genes near the ends of chromosomes. Without telomeres, the genomes would progressively lose information and be truncated, that is, shortened after each cell division. Thus, the telomeres can be considered to be disposable chromosomal buffers that help maintain stability of the genes but are gradually consumed during repeated cell divisions. Each time a cell divides, an average person loses 30 to 200 base pairs from the ends of that cell's telomeres. In human blood cells, the length of telomeres ranges from 8,000 base pairs at birth to as low as 1,500 in older people. Eventually, when the telomeres shorten to a critical length, the chromosomes become unstable and the cells die. This process of telomere shortening is believed to be an important reason for some of the physiological changes associated with aging. Telomere erosion can also occur as a result of diseases, especially those associated with oxidative stress and inflammation. In some cells, such as stem cells of the bone marrow or skin that must be replenished throughout life, or germ cells in the ovaries and testes, the enzyme telomerase adds bases to the ends of the telomeres so that many more generations of cells can be produced. However, telomerase activity is usually low in most cells of the body, and after many generations, the descendant cells will inherit defective chromosomes, become senescent, and cease dividing. This process of telomere shortening is important in regulating cell proliferation and maintaining gene stability. In cancer cells, telomerase activity is abnormally activated so that telomere length is maintained, making it possible for the cells to replicate over and over again uncontrollably. See figure 1.8. Some scientists have therefore proposed that telomere shortening protects us from cancer and other proliferative diseases. Regulation of cell size. Cell size is determined almost entirely by the amount of functioning DNA in the nucleus. If replication in the DNA does not occur, the cell grows to a certain size and thereafter remains at that size. Cell differentiation. A special characteristic of cell growth and cell division is cell differentiation, which refers to changes in the physical and functional properties of cells as they proliferate in the embryo to form the different body structures and organs. The following description of an especially interesting experiment helps explain these processes. When the nucleus from an intestinal mucosal cell of a frog is surgically implanted into a frog ovum from which the original ovum nucleus was removed, the result is often the formation of a normal frog. This experiment demonstrates that even the intestinal mucosal cell, which is a well differentiated cell, carries all the necessary genetic information for development of all structures required in the frog's body. Therefore, it has become clear that differentiation results not from loss of genes but from selective repression, that is, suppression of different gene promoters. In fact, electron micrographs suggest that some segments of DNA helixes that are bound around histone cores become so condensed that they no longer uncoil to form RNA molecules. One explanation for this is as follows. It has been supposed that the cellular genome begins at a certain stage of cell differentiation to produce a regulatory protein that forever after represses a select group of genes. Therefore, the repressed genes never function again. Regardless of the mechanism, mature human cells each produce a maximum of about 8,000 to 10,000 proteins rather than the potential 20 to 25,000 or more that would be produced if all genes were active. Embryological experiments have shown that certain cells in an embryo control differentiation of adjacent cells. For example, the primordial chorda mesoderm, that is in the fetus, early stages, is called the primary organizer of the embryo because it forms a focus around which the remainder of the embryo develops. It differentiates into a mesodermal axis that contains segmentally arranged somates and as a result of inductions in the surrounding tissues causes the formation of essentially all the organs of the body. Another instance of induction occurs when the developing eye vesicles come into contact with the ectoderm of the head and cause the ectoderm to thicken into a lens plate that folds inward to form the lens of the eye. Therefore, a large share of the embryo develops as a result of such inductions, with one part of the body affecting another part and this part affecting still other parts. Thus, although our understanding of cell differentiation is still hazy, we are aware of many control mechanisms whereby differentiation could occur. Apoptosis, programmed cell death. The many trillions of the body's cells are members of a highly organized community in which the total number of cells is regulated not only by controlling the rate of cell division but also by controlling the rate of cell death. When cells are no longer needed or become a threat to the organism, they undergo a suicidal programmed cell death or apoptosis. This process involves a specific proteolytic cascade that causes the cell to shrink and condense, disassemble its cytoskeleton, and alter its cell surface so that a neighboring phagocytic cell, such as a macrophage, can attach to the cell membrane and digest the cell. In contrast to programmed death, cells that die as a result of an acute injury usually swell and burst due to loss of cell membrane integrity, a process called cell necrosis. Necrotic cells may spill their contents, causing inflammation and injury to neighboring cells. Apoptosis, however, is an orderly cell death that results in disassembly and phagocytosis of the cell before any leakage of its content occurs, and neighboring cells usually remain healthy. Apoptosis is initiated by activation of a family of proteases called caspases, which are enzymes that are synthesized and stored in the cell as inactive pro-caspases. The mechanisms of activation of caspases are complex, but once activated, the enzymes cleave and activate other pro-caspases, triggering a cascade that rapidly breaks down proteins within the cell. The cell thus dismantles itself, and its remains are rapidly digested by neighboring phagocytic cells. A tremendous amount of apoptosis occurs in tissues that are being remodeled during development. Even in adult humans, billions of cells die each hour in tissues such as the intestine and bone marrow and are replaced by new cells. Programmed cell death, however, is normally balanced by formation of new cells in healthy adults. Otherwise, the body's tissues would shrink or grow excessively. Abnormalities of apoptosis may play a key role in neurodegenerative diseases such as Alzheimer's disease, as well as in cancer and autoimmune disorders. Some drugs that have been used successfully for chemotherapy appear to induce apoptosis in cancer cells. Cancer. Cancer may be caused by mutation or by some other abnormal activation of cellular genes that control cell growth and cell mitosis. Proto-oncogenes are normal genes that code for various proteins that control cell adhesion, growth, and division. If mutated or excessively activated, proto-oncogenes can become abnormally functioning oncogenes capable of causing cancer. As many as 100 different oncogenes have been discovered in human cancers. Also, present in all cells are anti-oncogenes, also called tumor suppressor genes, which suppresses the activation of specific oncogenes. Therefore, loss or inactivation of anti-oncogenes can allow activation of oncogenes that lead to cancer. For several reasons, only a minute fraction of the cells that mutate in the body ever lead to cancer. First, most mutated cells have less survival capability than normal cells and they simply die. Second, only a few of the mutated cells that survive become cancers because most mutated cells still have normal feedback controls that prevent excessive growth. Third, cells that are potentially cancerous are often destroyed by the body's immune system before they grow into a cancer. Most mutated cells form abnormal proteins within their cell bodies because of their altered genes, and these proteins activate the body's immune system, causing it to form antibodies or sensitized lymphocytes that react against the cancerous cells, destroying them. In people whose immune systems have been suppressed, such as in persons taking immunosuppressant drugs after kidney or heart transplantation, the probability that a cancer will develop is multiplied as much as fivefold. Fourth, the simultaneous presence of several different activated oncogenes is usually required to cause a cancer. For example, one such gene might promote rapid reproduction of a cell line, but no cancer occurs because another mutant gene is not present simultaneously to form the needed blood vessels. What is it that causes the altered gene? Considering that many trillions of new cells are formed each year in humans, a better question might be to ask why all of us do not develop millions or billions of mutant cancerous cells. The answer is the incredible precision with which DNA chromosomal strands are replicated in each cell before mitosis can take place, along with the proofreading process that cuts and repairs any abnormal DNA strand before the mitotic process is allowed to proceed. Yet, despite these inherited cellular precautions, probably one newly formed cell in every few millions still has significant mutant characteristics. Thus, chance alone is all that is required for mutations to take place. So, we can suppose that a large number of cancers are merely the result of an unlucky occurrence. However, the probability of mutations can be greatly increased when a person is exposed to certain chemical, physical, or biological factors, including the following: 1. The ionizing radiation, such as X rays, gamma rays, particle radiation from radioactive substances, and even ultraviolet light, can predispose individuals to cancer. Ions formed in tissue cells under the influence of such radiation are highly reactive and can rupture DNA strands, causing many mutations. 2. Chemical substances of certain types may also cause mutations. It was discovered long ago that various aniline derivatives are likely to cause cancer, and thus workers in chemical plants producing such substances, if unprotected, have a special predisposition to cancer. Chemical substances that can cause mutation are called carcinogens. The carcinogens that currently cause the greatest number of deaths are those in cigarette smoke. These carcinogens cause over 30% of all cancer deaths and at least 85% of lung cancer deaths. 3. Physical irritants can also lead to cancer, such as continued abrasion of the linings of the intestinal tract by some types of food. The damage to the tissues leads to rapid mitotic replacement of the cells. The more rapid the mitosis, the greater the chance for mutation. 4. Hereditary tendency to cancer occurs in some families. This hereditary tendency results from the fact that most cancers require not one mutation but two or more mutations before cancer occurs. In families that are particularly predisposed to cancer, it is presumed that one or more cancerous genes are already mutated in the inherited genome. Therefore, far fewer additional mutations must take place in such family members before a cancer begins to grow. 5. Certain types of oncoviruses can cause various types of cancer. Some examples of viruses associated with cancers in humans include human papillomavirus (HPV), hepatitis B and hepatitis C virus, Epstein-Barr virus, human immunodeficiency virus, human T-cell leukemia virus, Kaposi sarcoma associated herpes virus, and Merkel cell polyoma virus. Although the mechanisms whereby oncoviruses cause cancer are not fully understood, there are at least two potential ways. In the case of DNA viruses, the DNA strand of the virus can insert itself directly into one of the chromosomes, thereby causing a mutation that leads to cancer. In the case of RNA viruses, some of these viruses carry with them an enzyme called reverse transcriptase that causes DNA to be transcribed from the RNA. The transcribed DNA then inserts itself into the animal cell genome, leading to cancer. Invasive characteristics of the cancer cell. The major differences between a cancer cell and a normal cell are as follows. 1. The cancer cells does not respect usually cellular growth limits because these cells presumably do not require all the same growth factors that are necessary to cause growth of normal cells. 2. Cancer cells are often far less adhesive to one another than are normal cells. Therefore, they tend to wander through the tissues, enter the bloodstream, and be transported all through the body, where they form nidai, that is, center for growth, for numerous new cancerous growths. 3. Some cancers also produce angiogenic factors that cause many new blood vessels to grow into the cancer, thus supplying the nutrients required for cancer growth. Why do cancer cells kill? Cancer tissues competes with normal tissues for nutrients. Because cancer cells continue to proliferate indefinitely, with their numbers multiplying every day, cancer cells soon demand essentially all the nutrition available to the body or to an essential part of the body. As a result, normal tissues gradually sustain nutritive death. Some cancers cause disruption of vital organ functions. For example, a lung cancer might replace healthy tissue to the extent that the lungs cannot absorb enough oxygen to maintain tissues in the rest of the body. So, dear viewers, this is the end of part 4 of the 4-part series on the genetic control of cell function in Medicine for Common Man series. Thank you for watching the video.